Hi everyone, today we will cover RNA-seq. In the lab we analyze RNA sequencing all the time for all of our manuscripts, and here you will get everything you need to do a complete analysis. I will first show you an outline of the different steps. I will then show you how and where to download all the raw data. I will show you how to install every single program in the pipeline, and in the process you will be looking over my shoulder how to execute every single command. To go from FASTQ files with RNA reads to a table of differentially expressed genes. Let's get into it. Okay, this is what our pipeline would look like. So first we are going to download a bunch of FASTQ files from control 1 to control 3, and the same for the experimental FASTQ files, experimental 1, 2, and 3. And we also are going to download the reference FASTA file that we are going to map all of these reads. So the first thing that we are going to do is the mapping, and this will lead us to this file. So we'll have control 1.sum file and all down to the experimental 3 sum file. Then we are going to do a conversion of these files from sum to BAM. So we have the control 1 BAM all the way to the experimental 3 BAM. Then we are going to take these and we are going to quantify the reads, taking into account the read count, the transcript length, multiple mappings, etc. So we will do the quantification. That will generate this bunch of files for control 1, quant 5, and all the same for all the experimentals until experimental 3. We'll have quant files. And finally, we are going to do our DEseq that will leave us with this DG table where we have like the genes and we have the fold change, the Q value for gene A, B, Z is sometimes change, sometimes things change, and the Q value ranges. Hey, if you need a refresher on the basic bioinformatic files, check out the video on the card above. And with that out of the way, let's first start downloading the data. We will be using the data in the DEseq tutorial example. So for the FASTQ files, just Google the run name, go to SRA and download the data from there. And you will need to repeat this process for each FASTQ file. For the reference Transcriptome, we go to GeneCont website. We download the FASTA reference and the metadata file linking transcript symbols and gene symbols. Now let's download all the software. For that we will use Docker. Docker itself is trivial to download and install. So first we activate Docker and then we pull all the images that we need. So I already have the images with the software on my computer, but I will show you how to download them in your Docker. Hey, this is deceptively simple. Just follow along and you will be surprised at how it works. For the mapping, we will be downloading or pulling the following Minimap2 image from biocontainers. We copy this pull command. Note that we do not have Minimap2 among the current images. When I run the command, it downloads the image that now is among the images in my doc. For the conversion of BAM to SAM, we will use this Samtools image, and for the read quantification, we will use this Salmon image. You can download it with the same command, both images, essentially docker pull name of the image, colon, name of the tag, and that's it. We have now everything. For the DEseq, we need to download the DEseq and tximport packages from Bioconductor, and we do this as usual. That's all the software we are going to use. So let's go through all the steps of the pipeline, starting by the mapping. Okay, this is the end folder result that I generated. I will relaunch the different parts of the pipeline for one run so that you see how it works. To launch the Docker image with access to our files, we open a terminal and go to the directory with the files. I'm there now. As you can see, there is no Minimap2 installed. We need then to run the following line. Here we create the variable Minimap2 so that when we call Minimap2, this is actually what will be happening under. We will run Docker. With this v parameter, we will be giving Docker access to the folder we are now. Then we will call the image that we just downloaded. And finally, we ask to run Minimap2 inside that image. So now when we enter Minimap2, we get the Minimap2 help. Now we can run the minimap command of our interest. We use the SR parameter for short reads. Then we pass the reference transcriptome that we downloaded at the beginning. And we start with one of the 6 FASTQ files that needed to be downloaded. We ask the program to create the same file with the same name of the run for simplicity. Now the program is running. If we check our folder, the SAM file is created with 0 bits. And as the read starts to be mapped, the SAM file starts to increase in size. So we need to repeat the same for the rest of the files. Hey, check my SnakeMate playlist if you want to learn how to automate this whole process. The card is now on screen. Now that we have the SAM files, to convert them to SAM, we set up the Docker container as before, and we run the following Samtools line. We use the views function to call the SAM file generated, and we are giving the same name to the BAM file with the exception extension. So the program starts to run, the BAM file is generated originally with 0 bytes, and then starts growing as time passes. And once we have the 6 BAM files, we set up the Salmon image and we run the following line as we had been doing before. Then we launch Salmon with the quant function. We pass the reference transcriptome. This L parameter is to indicate the type of library. Then we give the BAM file and we ask to put the results in a folder with the same name as the run. Inside that folder, we'll have the quant files that we will be using for the next step. So the pipeline is launched, the folder is created, and the quantification begins. Now we end up with these folders with the run names containing those quant files. And the rest is done in RStudio. First, we upload the libraries that we are going to use, including DEseq, of course. Then we upload the metadata that we have in the text files, where we indicate for each run the condition. Here we are going to compare the runs in condition A to those in B. To convert the quantification from transcript-based to gene-based, we upload the gene symbol data that we downloaded at the beginning of the video. So we get for each transcript an associated gene symbol. Here we indicate where the quant files are. We do the conversion from transcript to genes using the text import function. With that, we create the DEseq DDS object, and finally we run the differential expression analysis. So here is the desired table. For each gene, we have the fold change between conditions and the associated Q value. Well, that's all. Subscribe if you like the content. And now that you have your differentially expressed gene, check out this video to get the gene ontology analysis in no time. I will see you there.